Hello, I have again come here with a new topic that is also related to biology. I would say it's not just biology, but the concept is about biochemistry, about nucleic acid chemistry. Okay, so basically the topic is nucleic acid. So I have tried to here show some few things. So today we are going to discuss it from the very beginning, from the very rudimentary concept. So what is nucleic acid? My question is, what is nucleic acid? Okay, what is nucleic acid? First we have to understand nucleic. So nucleic means present in nucleus, and acidic in the sense there may be some negative charge. Okay, because as we all know that where there is an acidic thing, it becomes a negative, like CH3COO, acid. Okay, negative charge comes from the acidic and positive charge comes from the basic part. So now inside the nucleus these nucleic acids are present. So what are the nucleic acids we have in normal cell? What are the nucleic acids are there? So nucleic acids are three types. I mean DNA and RNA. Sorry, that's not a three type, I'm sorry. DNA, RNA. So what is DNA, RNA? What is this DNA, RNA? The DNA is deoxyribonucleic acid. D for deoxy, R for ribo, deoxyribonucleic acid. So DNA. And you can easily understand that if DNA is deoxyribonucleic, then RNA is just ribonucleic. Okay. Now DNA, RNA. So these are the fundamental things of the cell, because due to the presence of nucleic acids the cells are alive. Why? Because DNA, RNA, these are the main things of life, because from DNA it becomes RNA, from RNA it becomes protein. Okay, now the protein composes everything, our muscle, every enzyme that we have in case of digestive system. Everywhere you just see any carrier protein, see the name is already protein. So in every there is an involvement of protein. So why ribonucleic acid, deoxyribonucleic acids are important, means DNA, RNA are important. These are the reasons. Now come to the part of DNA and RNA. Now what kind of molecule is this? Okay, means it's a protein, it's a what? Actually it's what is its composition? Okay, so we can say what is the structure of DNA? Structural composition. Deoxyribonucleic acid. both the nucleic acids composed of one sugar, two base, three phosphate. Now, if the sugar is present and the base is present, let me just draw the structure. The sugar, if I just take the easiest one, these are the bases. Okay, these are the bases. How many types of bases are there? 4 bases. Adenine, guanine, cytosine, and fourth, uracil. Also there is another one, it's called thymine. There are many different bases, but these are the bases that we find in RNA and DNA mostly. And sugar is all ribose sugar, that is 5 carbon, and phosphate we all know, PO4 3 minus. Okay, now let me draw something. So as we have understand the ribose sugar, so ribose sugar looks like this. Okay, this is the furanose form of ribose. Okay, furanose. Why furanose? How will you remember this is a furanose? F for furanose, F for 5. So carbon number is 5. Okay, so there are two types of sugar ring structure. One is pyranose, that is pyranose, and furanose. Okay, 6 carbon, 5 carbon. Now there are two terms. It's called, there are two terms. It's called nucleotide and nucleoside. What is nucleotide? Nucleotide is if in the nucleic if there is combination of sugar plus phosphate plus base. And what is a nucleoside? Just sugar plus base. So means only the difference is the absence of phosphate. In case of nucleotide there is phosphate, in case of nucleoside there is no phosphate. Just this is the, see the subtle differences. Now let me just rub all these things. So we have talked about the sugar, how the sugar is. Now let's talk about the bases. How many kinds of bases are there? So there are basically two types of bases. Purine and pyrimidine. As I have written here, adenine, guanine, cytosine, uracil, thymine. Among them, purine bases are adenine and guanine, and pyrimidines are cytosine, uracil and thymine. Okay. Now adenine and guanine, these are purine bases. Thymine, cytosine, uracil, these three are pyrimidine bases. Okay. Now the basic structure of pyrimidine and purine ring, let me just draw. Means suddenly why there is two categories, pyrimidine and purine. So there must be some similarity between adenine and guanine, that's why they are included in one category called purine. Purine has a common structure. This is the common structure of purine. Sorry. This is pyrimidine and the purine has double cyclic ring. Okay, now the double bonds are here. This is the typical purine. Okay, this is how the purine looks like. There is NH, okay, the nitrogen has bonded with the hydrogen. So this is the typical structure of purine and this is the typical structure of pyrimidine. How we number them? 1, 2, 3. I mean 1, 2, 3, 4, 5, 6. And in case of purine, how we number them? This starts from here. 1, 2, 3, 4, 5, 6. 7 starts from here, 7, 8 and this one is 9. Okay, this is the basic structure of pyrimidine and this is the basic structure of purine. Now we have discussed in under the category of purine there are adenine and guanine, and in the category of pyrimidine there is cytosine, uracil and thymine. So let's focus on the structure of first pyrimidine. Okay, then we will jump from pyrimidine to purine. So consider the structure of first cytosine. Okay, now cytosine contains what? We have to first write draw the basic pyrimidine ring. Okay, then there are subtle difference. There will be no bond because already the carbon has 4 bonds, 2 with oxygen and 1 with 2 other with hydrogens. So this is the structure of cytosine. Okay. Now if we just another H bonded with nitrogen and another bonded with nitrogen. Okay, let me draw it separately. So now if we draw the second is uracil, the basic structure is this. Only here will be the double bond, nowhere else double bond. Okay, so this is the uracil. Okay, and let me just erase all this. The last one is thymine, right? The thymine is just another methyl group. If we just add a methyl group, in uracil, so uracil plus 5th position methyl becomes, sorry, I have write down the vitamin B1, thymine. Okay, this is the thymine. So uracil with 5th position as the methyl group, this becomes the thymine. So these are the structure of the bases, and it is just the pyrimidines. Now let's come to the purines. Okay, now as we can see, there are two purines, adenine and guanine. So let's first draw the adenine, but before that the basic purine ring we have to draw, that is already given here. So this is the basic structure of purine. What is the addition? Just the addition is an primary amine group, and it becomes adenine. So adenine is basically at the 6 amino purine. We can write it as 6 amino purine. So as I was asking, now they come to the another one, that is the guanine. Okay, guanine is also almost same, but the easy trick is, if you can just learn the basic pyrimidine and purine structure, so you will be able to easily remember the different just by remembering the side chains. Okay, now here is the N H, here is double O, NH2 primary amine, and only the double bonds will be here, here and here. This is the structure of guanine. We can also call this 6-amino 6-oxypurine. Why 6-amino 6-oxypurine? The last one was 6-aminopurine because the whole structure is of purine and this is the amino at 6th position. And now why it is called the 2-amino 6-oxypurine? 2-amino means this is the 2-amino 6-oxy. So the name is 2-amino 6-oxypurine. Okay, because except the amino group and the oxy group, means the oxygen, all the things are same as the purine, normal purine ring, basic typical purine ring. Okay, so this is the different bases of the nucleic acid. And as we all know, what is a phosphate? PO4 3 minus. Okay, P double bond, okay, and sorry, this is the double bond with oxygen and another, because phosphate valence is 5, so the phosphate will be like this, and that's why PO4 and the 3 minus. So these are all the compositions of the the nucleic acid, the sugar, the base is very important, and the phosphate. So in the next session we will talk about the function and different things.